Introduction. At the outset, I would like to thank Dr. Sunita Agarwal for inviting me for delivering this talk. This talk of mine is a bit out of space because I'm neither going to talk on autologous DNA nor on stem cells. It will be on a prominent growth factor, namely vascular endothelial growth factor, that is VEGF, with reference to a neurodegenerative disease, namely amyotrophic lateral sclerosis. So from neurodevelopmental disorder, we are moving on to a neurodegenerative disorder. Vascular endothelial growth factor, as the name indicates, has a prominent role in angiogenesis and in associated areas of angiogenesis, so such as immunomodulation, vascular homeostasis, blood pressure regulation, thyroid function, etc. But most prominently, it has its function in vasculogenesis and angiogenesis, which are essential for formation of the blood vessels. So VEGF has several isoforms, A, B, C, etc. Each of these isoforms bind to a specific receptor, and binding of VEGF to its specific receptor activates the downstream events and ultimately facilitate angiogenesis. For example, VEGF binds to its specific receptor, VEGF-R2, and then activates the downstream pathway, namely phosphatidylinosytol 3 kinase pathway, AKT pathway, etc., which ultimately bring about cell survival, vascular cell formation, etc. So basically into the angiogenesis process. This particular factor is a hero when it comes to certain disorders like traumatic spinal cord injury, where it promotes nerve repair. But in certain conditions like tumor and diabetic retinopathy, it's a villain because it worsens the disease condition. Now, I said I'll be speaking about VEGF with reference to a neurodegenerative disorder. What has VEGF got to do with a neurodegenerative disorder, namely amyotrophic lateral sclerosis? Before we go into what VEGF has to do with ALS, let's know briefly what ALS is. We are all aware of the Ice Bucket Challenge. This Ice Bucket Challenge was initiated to bring in awareness regarding this neurodegenerative disorder, in which there is specifically the neurons which control our voluntary movement, the motor neurons, are degenerated. And this Ice Bucket Challenge was initiated to raise awareness regarding the disease and also to raise the funds for research, etc. So we also know about Stephen Hawking, the famous physicist, who recently passed away. He suffered from this disease. Just to understand the depth of how devastating this disease is, I would like to quote a few lines by an ALS patient. Quote unquote, "I am a person with ALS, searching, hoping, praying, learning and trying to understand what is slowly stealing my strength. The feeling of helplessness is debilitating. The feeling of my spirit leaving my body is frightening, yet peaceful. This is a cruel disease as it's slowly taking away my ability to walk. It's taking my voice, my ability to dress myself and feed myself." So here we understand how devastating this disease is. Any patient who gets this disease will die within 3 to 5 years, mainly because of the failure of the respiratory muscles towards the end of towards the terminal stage of the disease. This disease is also called as Lou Gehrig's disease, as named on behalf of the famous baseball player who suffered from this disease again. You'll get more insights regarding the disease, the clinical symptoms, etc. in the next session by Professor T. N. Satyaprabha. But as of now, what does amyotrophic lateral sclerosis mean? It means that it's atrophy of muscle fibers, that is A without, myo refers to muscle, and trophic is nourishment. So there is atrophy of muscle fibers because the motor neurons are degenerating. And lateral sclerosis refers to the hardening of the lateral column of spinal cord, which is caused because of degeneration of motor neurons, which are replaced by the glial cells. Now, what has the growth factor got to do with ALS? So this was because of a serendipitous discovery where VEGF has a role in hypoxia. Its expression increases during hypoxia. So researchers were focusing on a promoter region to which a hypoxia-inducible factor binds to this promoter region of VEGF gene. So they deleted the promoter region to which the hypoxia-inducible factor binds to, and then this particular transgenic mice, surprisingly in its adulthood, developed a motor neuron degeneration and symptoms similar to in ALS. In addition, there is another patient who was being treated for long term with VEGF receptor inhibitor developed ALS. Subsequent to these findings, there have been several studies which have been going on to understand the role of VEGF in ALS. It's been reported that the baseline levels in the CSF or the cerebrospinal fluid of ALS patients show reduced levels of VEGF. And also in mice, in ALS there are few mutations. Around 2% of the total ALS cases show mutation in a gene called superoxide dismutase or SOD1, when in the transgenic mice with mutation in SOD1 who also had mutation in VEGF showed much more severe symptoms and decreased life expectancy. And there have been several studies in these familial models where to see whether VEGF has been neuroprotective, and it is seen that around 30%, there is 30% increase in life expectancy of these familial ALS mice without causing any side effects, that too when it was administered after almost 50% of the neurons were lost. So till date, this has been one of the most effective therapies reported in the animal models, or to be more specific, the familial animal models of ALS. But in our lab, we focus on sporadic form of the disease where there is no known cause of mutation in any of the genes, and we do not know the cause of this, why the sporadic diseases occur, why the sporadic form of the ALS occurs, but it represents almost 90% of the cases. So what we do is we take the CSF or the cerebrospinal fluid of the ALS patients, and we have exposed the embryonic rat spinal cord cultures and a few of the motor neuron cell line to the ALS CSF samples, and also we've injected the CSF samples into rat neonates and adult rats to study the mechanism of action of the ALS CSF and to study the disease pathomechanism, specifically the sporadic ALS pathomechanisms. In this particular study, our question was whether VEGF has a neuroprotective role in sporadic ALS similar to what was seen in the familial ALS case, which represents only less than 10% of the cases. So to address this, we have chosen NSC34 motor neuron cell line because ALS involves degeneration of motor neurons. So we have chosen motor neuronal cell line, and we expose these cells to CSF for 48 hours, and subsequent to this, we have added recombinant VEGF protein and for 24 hours and looked at the neuroprotective effects. Here we can see that NSC34 cells on the right panel appears to be in two forms. That is one, the differentiated forms, which represent the mature motor neurons, whereas on the other side, which you see are the dividing cells, because these cells are fusion products of the neuroblastoma cells with the spinal motor neurons of mouse. Upon addition of ALS CSF, what we see on this panel here is that under normal conditions, we see that there are more differentiated cells, but upon addition of ALS CSF, there was reduced differentiation, and also we found cytoplasmic vacuolation in the cells. In addition to it, we found beating of neurites. All these features, that is presence of cytoplasmic coagulation, beating of neurites, and reduced differentiation hints towards the toxic effect of ALS-CSF. Does VEGF have any neuroprotective effect? So, indeed, we found that VEGF had neuroprotective effects because all these reduced differentiated cells were seen to be well differentiated or the differentiation enhanced in the presence of VEGF, and there was the vacuolation seen and the beating of neurites which were seen in the presence of ALS-CSF were reduced upon addition of the exogenous supplementation of VEGF. We then wanted to look at the survival, whether VEGF has any survival effect. So we found that we labeled these cells for tunnel and here. So under normal conditions, we can see that there are hardly any tunnel positive cells, but in the presence of ALS-CSF, we could see several tunnel positive cells and also enhanced expression of caspase-3, indicating that ALS-CSF is indeed toxic and causing cell death. But in the presence of vascular endothelial growth factor, we found reduced number of tunnel positive cells and also reduced caspase-3 expression, and it also facilitated enhanced differentiation, suggesting that VEGF indeed has a neuroprotective effect. We also looked at its possible role on the necrotic form of cell death. So we collected the supernatant medium. Whenever there is any necrotic cell death, the cells release their contents into the surrounding medium. So we looked into the LDH or lactate dehydrogenase activity in the supernatant medium, and we found that the LDH activity was reduced in the cells cultures which were supplemented with VEGF. We then looked at the expression of cholinesterase transferase, that is in green what you see. Cholinesterase transferase is an enzyme which is involved in synthesis of acetylcholine, the neurotransmitters of the spinal motor neurons. Whenever the acetylcholine neurotransmitter is released, it facilitates contraction of muscle fibers. So for proper functioning, CHAT expression is very crucial. So what we found is, on one hand, in the presence of ALS-CSF, the CHAT protein expression was reduced, and also what you see in red is the expression of the phosphorylated neurofilaments. Neurofilaments form an axonal path for the transport of crucial ingredients in addition to CHAT protein. So we found that in the presence of ALS-CSF, there was aggregation of phosphorylated neurofilaments. And wherever there was aggregation of phosphorylated neurofilaments, we found that the CHAT protein was also accumulated, indicating that there is ALS-CSF is causing aberrant phosphorylation neurofilaments and leading to axonal transport deficits or defects. Subsequently we found that these aggregates were ubiquitinated, indicating that they are targeted for degradation. Upon supplementation of exogenous VEGF, what we found is that this aggregation of phosphorylated neurofilaments was reduced, and also the expression of CHAT protein was enhanced. And as you see here, quantitatively also, we found that the CHAT protein expression is significantly enhanced, and we also looked at the calcium binding protein expression, which was found to be downregulated in the presence of ALS-CSF, was restored back to normalcy. In view of all these findings, we looked into the ultrastructure of these cells, and what we found is, in the presence of ALS-CSF, we could see presence of apoptotic bodies, and also that the mitochondria were either swollen or shrunken. But these observations were reversed in the presence of VEGF. So VEGF was indeed neuroprotective, and what we saw is, in the presence of ALS-CSF, there were nuclear membrane dysmorphism and presence of stress granules, what you see in the arrowhead in A, and in the presence of VEGF, these stress granules were reduced, although not completely reversed, but the number of stress granules were reduced. Formation of stress granules is two other proteins play a major role in formation of these stress granules and are reported to be altered or mutated in ALS. Those are TDP43 and FUS proteins. TDP43 and FUS are majorly RNA binding proteins which are localized to the nucleus and not the cytoplasm, which is, as you see in NC, the first panel. So TDP43 is green and FUS is red in color. But upon addition of ALS-CSF, there is a mislocalization from nucleus to cytoplasm, and once we supplemented these cultures with exogenously supplemented them with VEGF, we found that the mislocalization was reduced to a greater extent. So we wanted to see what's happening to the expression of VEGF per se and also the VEGF-R2 receptor. The VEGF expression as well as VEGF-R2 expression were significantly reduced in the presence of ALS-CSF, but upon supplementation with VEGF, it was restored back. Several other studies have also reported the neuroprotective role of VEGF, that VEGF protects spinal motor neurons against glutamate-mediated excitotoxicity, and it also has neurotrophic effect, and it read not only on the neurons, but it also has protective effects in terms of reducing the astrogliosis and also taking the microglia to a protective lineage or protective form, that is the M2 form. So, in view of all these findings, VEGF has already entered clinical trials. At present, it is in phase 2 trial where its safety and tolerability is being looked into. So the take-home message of this study is that VEGF effectively reverts the sporadic ALS-CSF induced neurodegenerative changes in NSA34 cells, and it's a promising therapeutic agent to treat not just the familial form but also the sporadic form of ALS, and VEGF plays a multitasking role to induce this neuroprotection. Sorry, I had to rush through because I was seeing quite a few of you are hungry and desperately want to walk out. Thank you very much.